recording this, and hopefully you've been able to find the recordings of our sessions. So if you want to go back and look at anything, you had the ability to do that. I've been saving the recordings as YouTube videos in the modules. So for like today, this recording will be placed in module 15, and typically I post it on Blackboard at the very bottom. So you just scroll down, scroll down, scroll down, and you'll eventually get to it. All right, so let me share my screen with you, and we'll go over some announcements, make sure everybody understands what needs to be done over the next, well, between now and the end of the semester, which is next week, and then we'll go from there. Okay, so yesterday I went and I graded pretty much everything, and I added everything to Blackboard. I updated your current grades, and I updated your current points, and that includes your take-home exam. So you should be able to log into Blackboard and look at your take-home exam and see the correct answers, the answers you submitted, and the points you received. So it would be equivalent to me handing back your exam. You should be able to see everything, and it has been added to your grade. I also went and I updated the pre-labs. I updated the lecture quizzes, and I dropped your 2 lowest lecture quizzes. I had not updated all of your online assignments, including your exam 3 preparatory assignments. That's been done. The only thing I have not done is the digestive disorders and diseases activity, which is up to 5 points. So I plan to do that later today. And so hopefully by the end of the day, certainly by Friday, or by Saturday, I should say, your grade will reflect everything that has been turned in, every exam and quiz that you have completed. The only thing that will not be included is your final exam, which you'll be taking next week. And I always add in the extra credit at the end. You should be able to see your total of extra credit, but I add the extra credit last. I like for students to sort of forget that it's out there, and then it's sort of like the money that you find in your pocket that you didn't expect, right? You're always happy about finding extra money. So, so your current grade should reflect everything except the digestive disorders and diseases. The practical is currently open, and it will close on Friday at midnight. It is the same format as the previous practical. There are 50 questions. You have a time limit of 42 minutes. There's no backtracking, and the Quizlets are no longer available. I literally took them down about 5 minutes ago, 5 to 10 minutes ago. Make sure when you are completing the practical that you read the questions carefully. And some of the quizzes, students would give answers which didn't match the questions. Like there was a question where I was pointing to the chordae tendineae, and I specifically said identify the string-like structures, and many students responded valves, or they responded a specific valve, and I was reporting the 2 valves, and so it wouldn't make sense that I was asking for the name of 1 valve when I had arrows going to both. And if you would read the question carefully, you would know, hey, he's asking for the chordae tendineae, not the mitral valve or the bicuspid valve. Also, if it's an artery or vein, you must include at least A for artery and V for vein. You don't have to write the whole word out, but you have to at least include that. And if it's one of those few arteries where you have to use left and right, like left and right coronary artery, then you must include those. Okay? Your final exam will be next week. Now, typically, I open the exams on Thursday and they close on Friday. My intent is to open it as early as Wednesday. I might open it earlier. Whenever I have it done, I'm gonna post it, and so you can start as early as I can make it available. But I will commit to definitely having it available on Wednesday, and then it will remain open until Friday at midnight. Please don't wait till the last minute. Now the exam has two components to it. The first component, well, I should say one component, will be the new material. The new material will basically cover the respiratory system, the digestive system, and the urinary system, which is what we're currently completing. The format's going to be the same as the previous exam. You're going to have 2 parts. Each one will have 25 true-false questions, 25 multiple-choice questions. Okay? Actually, that's not exactly how I've been doing it. Actually, I think I've had 50. How have I been doing that? Those numbers may not exactly be right in terms of the number. Maybe they are. Is it 50 each? I'll have to go look at that. But anyway, you're going to have equal numbers of true, false, and multiple choice for both of those parts. I thought they added up to 100, but I'll go and check that. Anyway, it's going to be the same format as exam 3. Exactly the same, no different. You will also have a comprehensive component. Basically, there will be 50 multiple choice questions, and they will cover all of the other chapters. Chapters 1 through 3, 4 membranes only, because the integumentary system is covered on the take-home exam. 5 through 7, and then 9 to 11. Basically, everything that was covered on exam 1, 2, and 3. Okay? Nothing from the take-home exam will be on the comprehensive exam. So this module is all about preparing you for the final exam. And to prepare you for the final exam, I have prepared some study activities for you to complete. If you complete the study activities for 2 chapters, you'll get 4 points. And if you complete the activities I've written for a 3rd chapter, then you'll get 2 points of extra credit. And then if you collaborate with another student, get their signature, you can get 2 additional points for extra credit. So you can get, a minimum of 4 points would be 100% of this assignment. But then you can get an additional 8 points of extra credit. Oh, by the way, this is due Thursday at 8 a.m. Even though I might be opening the exam earlier, like, well, definitely Wednesday, but even earlier, the study activities won't be due until Thursday at 8 a.m. And I'm flexible on the exact time, right? If they're not in exactly at 8 a.m., that's not a problem. Oh, make sure that you're uploading them to Blackboard. Okay. There's also a final exam self-assessment, just like all the previous self-assessments. Basically, you hit 75% or greater, and you'll get 2 points of extra credit. This only covers new material. It does not cover the old material. I did make the previous self-assessments available to you, but they're for you to practice, but they're not going to provide you with extra credit or anything like that. And this is due, again, 8 a.m. Thursday, May 21st. One thing that I didn't think of until earlier today, and therefore it's not on your official module for this week, and that is, I would like for you to complete a survey on the instruction that I have provided to you. Basically, it's a course evaluation. We are required to ask our students for an evaluation, and honestly, it's the only way that I can know what I'm doing right, what I'm doing wrong, and the steps I need to do to improve myself. It is 100% anonymous, so I want you to be very candid in your answers. I don't want you to feel that to hold back because you're afraid of like some retribution from me or something like that. I can tell if you have completed it, but I cannot know what answers you gave unless you identify yourself by typing in something that identifies you, like your name or something like that, or if I know something about you that you shared with me or something. There's no way I know who you are. So please complete that survey of student, your opinion of instruction. It's going to be available all the way up until June 1st. So there's plenty of time for you to do it. The earlier the better, so you don't forget. But that is very important. So I'd ask that you complete that. Okay. Oh, and I'll give you a point of extra credit if you do, because I can tell if you've done it, but I can't tell what your answers are. So I'll as a little carrot, I'll give you a point of extra credit if you will do that. Okay. Before I move on, let me ask if there are any questions about any of that I shared with you before we jump into today's material. All right, pretty good turnout for our last day, I would say. All right, the oh so silent class. I hope you won't be this silent through the entire session. Let's see here. Let me go back to my slides. Okay, so basically what I'm going to do is what I've done for the most part the entire semester, which is I'm using the muddiest points that you have indicated to focus our lecture time together. All right, and so I was a little surprised, honestly, by the muddiest points. The muddiest points, what you indicated were your muddiest points, seem to be like the most sort of cut and dry, easiest concepts in this chapter, and then the concepts that I thought were a little more complicated, no one had any questions about, such as the formation of urine. We'll be going over the formation of urine anyway, because I don't believe you. I will have to admit to a certain extent that you're oh yeah, I understand urine formation completely. And that, if you don't understand anything else, I want you to leave this module understanding the basics of urine formation. I mean, how can you understand the urinary system if you don't know how urine is made? So we'll definitely be looking at that. But I will definitely address your muddiest points today. And looking at the first screencast, there was questions about hydronephrosis, so we will address that, and the clinical significance of the trigone, and the learning objective explain the clinical significance of the trigone is almost as long as the explanation I'm going to give you. It's really not that complicated, but I will make sure that you understand the clinical significance of the trigone. Okay, so let's look a little bit at the anatomy of the urinary system. It's not a lot to it, at least not that you have to know. Here we have our kidneys, which are located in the abdominal region, and those kidneys produce urine. And then you have a ureter that then moves that urine inferiorly down to the urinary bladder, where it is stored. Now, the kidneys are constantly making urine. They make urine 24/7, while you're asleep, while you're awake, while you're walking around, while you're sitting, etc. So they're constantly making urine, and that urine is moving down the ureters. There are some cases where a kink, for lack of a better term, can form in that ureter. And if that happens, it's like having a kink in a hose. The urine backs up into the kidney, and then you literally have water on the kidney, and that's called hydronephrosis. This could be due to someone who goes on a crash diet and loses a lot of weight, and the fat that normally surrounds what's called the adipose capsule that normally surrounds the kidney is eroded, and the kidney sort of falls out of its normal position, and it puts a kink in the ureter. It could be due to something that grows in the ureter, but the bottom line is if there's some type of blockage that prevents the urine from moving down into the bladder, and so it backs up into the kidney. That's what hydronephrosis is. The second question was about the trigone. So the trigone is this sort of imaginary triangle that we can draw in the urinary bladder between the urethral orifices. That's basically the openings where the ureters enter the urinary bladder, and the opening to the urethra, which is the tube that allows urine to leave the urinary bladder. So if we draw these lines between those three locations, the imaginary triangle that is formed is called the trigone. The clinical significance of the trigone is that usually when one has a urinary tract infection, it usually starts here in the trigone, and that's because the source of infection is usually the urethra, or at least the bacteria in the outside world enter through the urethra and then they get into the trigone here. And while we're talking about urinary tract infections, let's talk about why we see differences in urinary tract infections between men and women, and it has to do with the location of the opening of the urethra in women compared to in men. So first of all, there's a difference in length of the urethra. In women, it tends to be very short, only about an inch long. And it is relatively close to the anus. Now, your feces is about 50% bacteria. So it is always a potential source of infection. Because of its proximity to the urethra, women tend to be more susceptible to urinary tract infections. It's also why it is important for women to, when they wipe after defecation, that they wipe front to back. Right? Women know this, but guys probably don't know this. So guys, right? If you have a daughter, right, wipe front to back. Okay, not back to front. Us guys, we can go either direction. It really doesn't matter, because there's a significant distance between our urethra and our anus. The urethra does tend to be longer in men for obvious reasons, because it has to go completely through the penis and up into the bladder. That makes implanting a catheter a little more involved and a little more invasive for men than it does women. So that's a significant difference there. Also notice that the urethra passes through the prostate, and as men age, we're more likely to have an inflamed prostate, and that can cause problems with what's called urinary retention, where it prevents water from urine, from moving down the urethra. Okay, but men are less likely to get urinary tract infections because the distance between the urethra and the anus is much greater. We're less likely to have contamination, fecal contamination. And even if one is practicing good toiletry habits, just that proximity, that closer proximity, women are more likely to have a urinary tract infections. Okay, any questions about any of that before moving on. It's pretty cut and dry. That's all I want you to know about the trigone. That typically, if there's a urinary tract infection, that's where we see it, or at least that's where it starts. And I want to make sure you understood hydronephrosis. Well, if there aren't any questions, let's move on to the nephron. Nephron's very important. It is the basic unit of the kidney. All of the nephrons together basically are responsible for the performance of the kidney. And the kidney, its main function is to produce urine. The muddiest points that you indicated were describe the flow of blood through the nephron and to compare and contrast cortical and juxtamedullary nephrons. Here is where I was a little surprised that these were muddiest points, because the flow of blood through the nephron, I feel anyway, is pretty straightforward, as is the difference between the cortical and the juxtamedullary nephrons. At least that was my perception. So this surprised me a little bit, but perhaps what was not clear was exactly what you were expected to know. So we're going to go through that now, and then I'm going to ask if there are any questions. Okay, so this is a frontal section of the nephron. First, I want to point out two regions of, this is a frontal section of the kidney. This is the frontal section of the kidney. I'm sure you knew that before I said it's the frontal section of the nephron. But this is the frontal section of the kidney. All right. First, I want to point out two general regions of the kidney. One would be the renal cortex. The outer portion of the kidney is the renal cortex. Cortex means bark, like the bark of a tree. It's on the outside. So whenever you hear the word or read the word cortex with respect to anatomy, always refers to the outside. Deep to the cortex is the medulla, and the medulla is thrown into these pyramid-shaped structures, and we just call them medullary pyramids. Right, so here's the renal cortex, and in the renal cortex we have cortical nephrons, and in the medullary pyramids, or actually I should say sort of spanning the border between the cortex and the medulla, or the medullary pyramids, we have the juxtamedullary nephrons. So here's a chunk of kidney here, and here you see the cortical region, and then here's the renal medulla. Cortical nephrons are completely found in the cortex. That's where they're found. That's why they're called cortical nephrons. Juxtamedullary nephrons are juxtaposed, shall we say, between the renal cortex and the medulla. Basically, they cross that border, and portions of that nephron are found in both. Thus, they're called juxtamedullary nephrons. Now, the significance of the juxtamedullary nephrons is that they have a greater ability to concentrate urine. So they play a very important role if you are not consuming sufficient amounts of water, right? Because they're responsible for helping conserve water. So that's one difference between them. The other difference is the location, and they're more cortical nephrons than they are juxtamedullary nephrons. Most of the nephrons are cortical nephrons. Fewer of them are just juxtamedullary nephrons, but the juxtamedullary nephrons have a greater ability to concentrate urine. Okay, that's the significance there. All right, let's talk about the flow of blood in the kidneys. Now, this image comes from your book, and what I tried to do, and maybe this was a little more confusing than I had expected it to be or had meant for it to be. And basically, what I was trying to do in this slide was identify what I thought was important for you to know and what you could pretty much ignore. Okay, so let's go through this. So first, of course, you know that all oxygenated blood leaving the heart leaves by the aorta, right? So and you also know that aorta descends into the abdominal region, and then there's a renal artery that comes off the aorta leading to the kidney. So this aorta, renal artery, you should already know from lab, right? That's nothing new. But just to put it in context, this would be the renal artery right there. Right, what's not shown here is the abdominal aorta, but that's the renal artery, and that renal artery is going to divide into smaller arteries and smaller arteries, interlobular arteries, lobar arteries, etc., which you don't have to know, which is why I didn't mention those other arteries. Those aren't important to understanding the formation of urine, right? So aorta, renal artery, you should know. All these other arteries, segmental arteries, lobar arteries, interlobar arteries, etc., those you can just ignore. They're not important to understanding the formation of urine. However, these blood vessels here are involved in the formation of urine, and they're found specifically in the nephron. So this little triangle here, what I was trying to bring to your attention is that, hey, you should know how blood flows through these arteries, that these blood vessels, that blood from the renal artery eventually makes it to the afferent arteriole, and then to the glomerular capillaries, then the efferent arteriole, and the peritubular capillaries. And I wanted you to pay special attention to these very specialized capillaries here. And notice that they're different from any capillaries we've talked about. And how are they different? Well, they're different because they are supplied and they are drained by arterioles. Up to this point, we've always described capillaries as being supplied by arteries and drained by veins. So this is special anatomy here. And if we've got some special anatomy, that means there's going to be some special physiology that's going to occur here with these glomerular capillaries, and indeed that's the case. Now let's look at this in context, right? So here is a nephron. This portion of the nephron is called the renal corpuscle, and then this portion is called the renal tubule. Let's look first at the renal corpuscle. Right, so here's a blow up of that renal corpuscle. Let me go back just to make sure I did that a little quick. Right, so where that box is, that's the renal corpuscle. We're going to focus on that first. So the renal corpuscle contains the glomerular capillaries that we just talked about. And we said the glomerular capillaries are fed by an afferent or afferent arteriole, and they're drained by a smaller efferent arteriole. So blood from the renal artery eventually makes it to the afferent arteriole, then into the glomerular capillaries, and then into the efferent arteriole. Blood leaving via the efferent arteriole is now going to move into the peritubular capillaries here. Now the peritubular capillaries, they start here, they wind all around the renal tubule there, and then they end there. Those peritubular capillaries are going to be very important in the formation of urine, particularly in the two processes called reabsorption and secretion. Later on, when we talk about how urine is formed, it's going to be important to understand the roles that all these blood vessels play in that process. And that's why I wanted you to know how blood flowed through these blood vessels. So we start with the afferent arteriole into the glomerular capillaries, and the efferent arteriole then leads to the peritubular capillaries. And then the peritubular capillaries eventually lead, the peritubular capillaries eventually lead back to the inferior vena cava. Well, excuse me, I should say, eventually lead to the renal vein and the inferior vena cava. Okay, so, oh my, where's, all right, so we start here with the aorta, then blood flows into the renal artery. Then through a series of arteries, which you don't really need to know, into the afferent arteriole, then from the afferent arteriole to the glomerular capillaries to the efferent arteriole, from the efferent arteriole to the peritubular capillaries. Notice the peritubular capillaries are your normal capillaries fed by an artery, drained by a vein, and then eventually the blood goes to the renal vein and then to the inferior vena cava. The blood vessels I boxed here, you should know simply from lab. The blood vessels I put in a triangle here, you should be familiar with because they are involved in the formation of urine, and you're expected to understand the formation of urine. So that's what I was trying to convey to you by boxing and triangulating, for lack of a better word, these specific arteries, is that, hey, these are the arteries that I think you should know, either because you've been exposed to them in lab or because they're involved in urine formation. Okay? All right, so let's take a pause here and let me open up the floor for questions. Because I want to have a conversation. I don't want to feel like I'm lecturing to you. I want to feel like we're having a conversation and you're having a chance to interact and ask questions. So for those of you who indicated these as your muddiest points, and it's probably pretty much everyone here because I've got like 13 muddiest points on these. Is this making sense? Do you still have questions? Or has this clarified things? Don't everybody talk at once. Okay. Katie says, it makes more sense to me. All right. Thank you, Katie, for your response. All right. How about someone else? Do you still have questions? Okay. Taylor says it's clear. All right. Clear as crystal. Does it like ting? She says it tings. All right. Okay, so well, please stop me if you have any questions whatsoever. Otherwise, I want to go on and talk about urine formation, even though no one indicated that urine formation was one of their muddiest points. But I just cannot believe that everyone is like, oh, 100% clear about how urine is formed. So what I would like to do is cover some of the parts of the learning activities that I emailed to you. So it looked like I emailed this to you yesterday and told you to bring it with you or have it available for our session today. So hopefully you have that at your fingertips. Because I want to go through urine formation, make sure you have a basic understanding of urine formation because it's very important. And again, if you're going to understand the urinary system, you sort of have to understand how the kidneys produce urine. Not to mention, I would think that since you're doing it all the time and you're seeing it all the time, you'd sort of have a bit of a curiosity to how's that stuff that I release from my body on the daily basis, how's that formed? Why is it dark at some times of the day and it's light at other times? How does that work? Okay, so here are the learning objectives with respect to urine formation. Explain the process of filtration, explain the process of reabsorption, explain the process of secretion, describe the normal characteristics of urine, and list substances normally found in urine. Now, because of time constraints, unless someone has a question, I'm going to focus primarily on explaining the process of filtration, reabsorption, and secretion. If you understand those, then you can certainly understand the normal characteristics of urine. And the substances that are normally found in urine. Okay, so this is a urine specimen. If you've ever provided a urine specimen, you know that it looks something like that. It's a nice little amber liquid, and the urine is made by the kidney. Where does urine come from? The components of urine are taken from where? Well, they're taken from the blood, and in fact, if we look, let's look specifically at the plasma. Doesn't the plasma look a lot like urine when you really think about it? They're both these amber liquids that are translucent, or you can see through. Though I should say not translucent. They're clear that you can see through normally. Well, it's no coincidence that they're similar in physical appearance, because urine is made from the plasma. In fact, we could describe urine as being an ultrafiltrate of plasma. Basically, what your kidney does is it removes the plasma from blood, minus the plasma proteins, and then it returns about 99% of the components of plasma back to the blood. And that 1% that it doesn't return is the urine. And that urine is high in, well, it's mainly water and with waste products and substances that the body doesn't need, maybe some, remnants of some poisons that you happen to consume in small quantities, of course, in the food that you have eaten and beverages that you have consumed, right? And that's what urine is. But urine comes from plasma, and that's what it looks like, plasma. So there are three processes that are involved in urine formation, and this sort of schematic here does an excellent job of illustrating that. Now, what you're looking at here on the right is a schematic of a nephron. Now, the images of the nephron in your book are quite busy, and so to sort of make it easier to understand, your author has constructed this figure to sort of illustrate the different parts of the nephron. Okay, so a nephron really looks like that, right? Okay, so very busy. So what do we? Oh, let me go back for a second. I'm sorry. All right, let me go back again. So this area up here, I think you can see this is called the renal corpuscle. And this illustrates the renal corpuscle. And this little tube here represents the renal tubule on the schematic, right? So very easy to follow. Now let's compare it to the nephron. So you can see here the renal corpuscle of the nephron. It includes the glomerular capsule, also called Bowman's capsule, and the glomerular capillaries. And then all of this out here with the peritubular capillaries winding around them is the renal tubule. Okay, so that's what we're looking at right here. We're looking at a less busier schematic of the nephron, because that's where urine formation occurs. Now, there are three processes involved in urine formation, so let's outline them here. The first occurs at the renal corpuscle, and that is filtration. Basically, anything small enough moves across the capillary wall and enters the renal capsule. Okay, and that's filtration. And what does that include? Well, basically, the plasma of blood minus the plasma proteins are filtered from the blood of the glomerular capillaries. Now think about that. So that's water. It's also nutrients like amino acids and glucose and calcium and phosphorus and iodine and all the different electrolytes. All that is filtered from the blood. Now, your digestive system spent a lot of time and energy digesting and absorbing those nutrients. So we literally don't want to piss away those nutrients. So that brings us to the second process of urine formation, which is going to occur in the renal tubule. And in this image, it occurs at B. So that filtrate moves into the renal tubule where reabsorption occurs. And in reabsorption, sometimes it's referred to as reclaiming. The kidneys reabsorb all the nutrients that the body needs. So a good percentage of water, glucose, amino acids, ions are going to be reabsorbed, and they're going to move from the renal tubule into the blood of the peritubular capillaries. The last process that is going to occur is secretion. So even though there were a large number of substances that were filtered from the blood in this first step, all the substances that the body needs to get rid of weren't filtered. There's still blood that passes from the renal corpuscle into the peritubular capillaries. And so the third process is going to serve to move those substances that the body doesn't need from the blood of the peritubular capillaries into the renal tubule. And that process is secretion. And it's basically reabsorption in reverse. So substances like, the breakdown products of that aspirin you took this morning because you had a headache. Or if you're taking a prescription drug, right, that's going to be broken down by your liver and other processes, right? Those products the body doesn't need, or the body's not going to recognize it needs, and it's going to be secreted from the blood of the peritubular capillaries into the renal tubule. Also, waste products like creatinine and urea, excess hydrogen ions, excess potassium ions, all of that's going to be moved from the blood of the peritubular capillaries into the renal tubule. So after those three processes, what remains is a substance that's mainly water with high levels of waste products that potentially, if they got to high enough levels, could be toxins, and substances that the body has more of than it needs, like hydrogen ions and potassium ions. And that's what we call urine. Okay, so that is an overview of the process of urine formation. So rather than lecture in detail on each of these processes, which is what I've provided in the screencast, what I thought we would do is go over the questions from that learning activity. So we're going to start with filtration. So here are the three questions that I have for you, and what I'd like to do is have you answer these questions. Take a moment to look at these questions, answer these questions, and let's go over them together. So filtration involves the move. So here are the three questions. Filtration involves the movement of substances from where to where. Second question, are plasma proteins filtered from the blood? Why or why not? What about glucose? Is it filtered from the blood? Why or why not? So let's take a couple of minutes. Where are we? 12:17. Let's go to 12:19. No, 12:20. 3 minutes. A minute per question. Right? 12:20 to answer these questions. And then I'm going to call on people to give me your responses. Okay, we got one more minute. Okay, so let's answer this first question. Filtration involves the movement of substances from where to where. And so what I'll ask you to do is raise your hand if you want to answer this question. And if you want to answer, if you're like, hey, I don't know both answers, if you know the first, that's fine. Just give us somewhere to start. So where, talk about filtration, where are the substances that are filtering? Where do they start? Use the little hand feature, hand raising feature to raise your hand, and then you can either type it in or you can answer orally. Come on, somebody wants to take this. You got this. Come on, come on. Well, I'll tell you what, I'll help you out. So it's going to start, substances start in the blood of the glomerular capillaries. And so where do they go? Oop. Obviously, I can't spell capillaries. Is that two L's? All right, here it is right here. So it's the blood. It's starting in the. We're filtering from the blood of the glomerular capillaries into where? Where is the substance going? Well, it's going into the glomerular capsule here. So it's starting in the glomerular capillaries. It's moving into the glomerular capsule. So when we talk about filtration, it's the movement of substances from the blood of the glomerular capillaries into the glomerular capsule. It's also called Bowman's capsule, although we're trying to get away from names of structures named after people. Okay? All right, so here's the next question. Are plasma proteins filtered from the blood? And if you say yes, why yes? And if you say no, why no? Well, if you said no, you are correct. And the reason why is they are too big. Substances about the substances have to be about the size of a small protein or smaller to be filtered, and proteins are too big. That's also why red blood cells and white blood cells aren't filtered from the blood. They're just simply too big to cross the walls of the glomerular capillaries. What about glucose? Is glucose filtered from the blood? Anybody want to try that one? Yes, Katie, thank you so much, Katie. Yes, it is filtered because it's small. Yes. Right, and if you're thinking, well, wait a minute, but you shouldn't have glucose in your blood, you are correct. However, it is found in the filtrate, as are amino acids and lots of other nutrients that you need. Okay, all right, so these questions were intended to help make sure you understand what the process of filtration is. Before we move to the next step, anyone have any questions about that, or is there any reason why? Do you have any questions about these answers, or these questions, or any segment of any portion of filtration before we move on to excuse me, before we move on to reabsorption? Okay, so after filtration, we have this filtrate that's now going to move into the renal tubule. Now this filtrate basically is plasma minus the plasma proteins. Anything small enough has been filtered from the blood. So it's basically plasma minus the plasma proteins because they're too big. That includes lots of substances that the body needs, and so now we're going to go through the process of reabsorbing those. Okay? All right, so here are our next questions. In general, when substances are reabsorbed, they are moved from where to where? All right, I'll give you the from where, and then you can fill in the to where. And then I want you to list some substances that are reabsorbed and where in the renal tubule does most reabsorption of solutes occur, right? So we're talking about going from the renal tubule to where? When we're talking about reabsorption. By the way, I guarantee you questions related to these will be on your exam. Okay, guaranteed. It's not even a possibly. Alyssa, very good. The peritubular capillaries. Peritubular capillaries. And unlike me, you can actually spell capillaries. Very, very good. So, all right, everyone, throw me some substances. You can just type it in that are going to be reabsorbed. What was, all right, water, very good. What else? Glucose, right? In fact, normally 100% of the glucose should be reabsorbed. All right, good. Sodium, all right, I need this. Trying to look at two screens here. No acids. All right, sodium. What about if you don't want to get osteoporosis? What would need to be reabsorbed? Calcium. Very good. And the list goes on and on and on. It'll be chloride, depending on the situation, it could be bicarbonate, all kinds of stuff. But the bottom line is the nutrients that the body needs, they're going to be reabsorbed because we don't want to lose those. Magnesium, yes. Right? And then there'll be others. Ooh, I've run out of my space. Okay. All right. Anyone know? So that renal tubule is composed of three components. You've got the you got the proximal convoluted tubule, the distal convoluted tubule, and between the two you have the loop of Henle. Anyone know where most reabsorption occurs among those three? Okay, which one? Tell me which one you think, Logan. Distal or proximal? Proximal, very good. All right, now they're talking to me. Distal convoluted tubule, proximal convoluted tubule. That's where most reabsorption occurs. And it's what's called obligatory, meaning it doesn't matter like how much glucose is in your urine. How much glucose was filtered or how much sodium was filtered. The same percentage is going to be reabsorbed, right? Very, very good. Okay, and it is these illustrations here where I go into where that occurs, right? This is the proximal convoluted tubule, loop of Henle, distal convoluted tubule. Most reabsorption occurs here at the proximal convoluted tubule. Very good, Logan. Very good. All right, and let's get to our last questions, which has to do with secretion now. So this is our third part. So, in general, when substances are, gosh darn it, my apologies. Where am I here? Okay. In general, when substances are secreted, they are moved from where to where, right? So we said that secretion is reabsorption in reverse. So substances are going to be moved from the peritubular capillaries. I still can't spell capillaries, can I? To where? Very good. Renal tubule. Awesome. And what are some substances that are going to be secreted that the body has no need of? Right, like drugs, waste products, right, poisons. And when I say poisons, and when the book talks about poisons, you do know that there are some very low levels of it could be certain minerals. You're correct, like nitrogen. You do understand that there's low levels of toxic substances in the food that we eat and the beverages that we consume. Now, hopefully, they're at such low levels they're not going to do us damage, and our kidneys are going to be able to get rid of them, right? But that's when we talk about poisons. That's what we're talking about. There's a small amount of arsenic in different foods. There's cyanide, believe it or not, in apple seeds. So if you've ever eaten an apple seed, you've taken some cyanide in, right? But because we have mechanisms to deal with those things, like our kidneys, to get rid of them, right? They don't usually cause us trouble unless they're above a certain amount. Okay, where in the renal tubule does most secretion occur? Give it to me again, Logan. You got this. Proximal convoluted tubule. All right. There's one location in the renal tubule where secretion does not occur. Anyone know where that is? So loop of Henle, although I've recently learned, again, we're trying to get away from eponyms named after people's names, and now they're calling it the renal loop, right? But there's no secretion that occurs there. It's all reabsorption. All right, cool. So those, again, are the three processes that are involved in reabsorption. In the production of urine. And if you understand those processes, then you'll understand why we find certain substances in urine normally and why you shouldn't find other substances in urine. Like you should never find glucose in urine. If you do, it means that the person has hyperglycemia. More likely, either they've consumed a meal that had a high level of glucose, excuse me, a high level of sugar, right? They could have just gorged down in candy, or they have untreated diabetes. And what's happening is so much glucose is being filtered that the kidneys just can't reabsorb it all, and you're getting some in the urine. Okay, before we leave, urine formation. Are there any questions? I want to make sure I answer any questions that you may have about urine formation. Alrighty, so really quickly, I want to cover urine storage and micturition. So, because we had a couple of questions about that. Well, the normal pH really, really varies. So the numbers, and when we get to acid-base balance, I'll explain why. But it can really vary quite a bit. So I'll get to that when we get to acid-base balance, if that's okay. So that urine is formed by those nephrons in the cortex and the medullary pyramids, and then that urine then moves through these calyces into the renal pelvis, and that renal pelvis leads to this muscular tube called the ureter, and that ureter descends all the way down to the pelvis and enters the urinary bladder, and urine moves down the ureter under the pull of gravity, and there is some peristalsis that occurs as well. So that's how urine makes its way from the kidney to the urinary bladder. All right, and you can see here the orifices where the urine enters the urinary bladder there at the two angles of the trichome. Kidneys produce urine continuously. In fact, over 24 hours, somewhere between 150 and 180 liters of plasma go through the glomerulus, right? Why do we have a urinary bladder? A urinary bladder basically stores urine. We're making urine constantly so that you can wear pants. We have a urinary bladder. The alternative would be we leak urine all day long and all night long, right? Okay, which would be an absolute mess. So we have this urinary bladder that stores urine until it's an appropriate time for us to void. Okay, urinary bladder is basically this muscular sac, and the walls of the urinary bladder are thick with this muscle. So it's basically a collapsible muscular sac. There's transitional epithelium lining the inner of the mucosa. And if you remember, transitional epithelium can sort of convert from simple to stratified depending on as it's stretched and recoils. Bladder can stretch quite a bit from, say, 2 inches to 5 inches when it's completely full. Okay, let's talk about the process of micturition or voiding, right? And it comes with many names: draining the lizard, going to see a man about a horse, or my favorite, knocking the dew off your lilies. But basically what we're talking about here is moving urine from the bladder through the urethra to the outside world. Urine leaves through the urethra, which is that little tube that connects the urinary bladder to the outside world. Notice it's got to pass through 2 openings. First, it's got to pass through the internal urethral orifice, and that is controlled by this internal sphincter muscle, which is smooth muscle. It also has to pass through the outer urethral orifice, and that is controlled by skeletal muscle. So that part is under voluntary control. So there's a part of micturition or urination that is under voluntary control and a portion that is under involuntary control. All right, okay, but when we talk about micturition or voiding, we're talking about urine moving from the urinary bladder through the internal urethral orifice and then through the urethra itself and through the external urethral orifice. All right, so it has to pass through the urethra. Okay. All righty, so let's go through the process. So what happens that leads to the micturition reflex? So as urine fills the urinary bladder, it causes the walls to stretch, and there are stretch receptors that detect that stretch, and they send impulses to the sacral spinal cord. That's where the reflex center for the micturition reflex is located. In response to that sensory input, motor output is sent to the smooth muscles of the urinary bladder, causing them to spasm. When that happens, the internal urethral sphincter allows urine to pass into the urethra. You feel that, and at the same time, sensory input is sent to your medulla oblongata. Those two things occurring, that urine entering your urethra and that sensory input going to your medulla oblongata, is what's responsible for that urge that you get to go. When you're like walking, you go, ooh, I gotta go to the bathroom, right? That's what's happening. Now, if it is in the appropriate time to void, then you will go to the bathroom and then you would relax that external urethral sphincter and voiding occurs, right? That's what happens when you go and relieve yourself. However, well, let's say you're like where you are now and you get that urge, right? And you're like, I'm in class, or you're riding in your car or whatever. Like, no, this is not a good time, right? So then you don't relax the external urethral sphincter. Eventually, those muscles will fatigue and the micturition reflex will subside. However, as you know, you will eventually get that urge to go again. And what's happening is urine is continuing to fill that urinary bladder, and eventually that cycle is going to start again, where sensory input sent to the micturition center, and then motor output sent back to the detrusor muscles, and they stop spazzing again. You have that whole series of events occurring again where you're like, oh gosh, I really gotta go. Okay, and eventually you will have to go. You can only suppress the micturition reflex for so long. Okay, any questions about the micturition reflex? And so hopefully this sort of shines some light on why it is like you gotta go, but if you can't go, after a while the feeling goes away, but then it comes back, and that cycle continues until you go, either voluntarily or involuntarily. Eventually we lose the ability to suppress that, right? I don't know if you've ever been in that situation, but eventually you got to go. The defecation reflex, unless you have diarrhea, you can suppress that to the point that you become constipated. You can't really suppress the micturition reflex, right? Eventually you will pee your pants if you don't void. So from water and electrolyte balance, there was one muddiest point that was indicated, and that was the effect of antidiuretic hormone on water output and water balance. So let's address that really quick. All right, so this is the nephron here. You can see that here you have the proximal loop of Henle, distal convoluted tubule, and the collecting duct. Now let's say that you're well hydrated. You're drinking lots of water. Well, as you know, when you're drinking lots of liquids, your urine you will tend to produce a lot of urine, and it will tend to be clear. The reason why that is because it's a dilute urine. If you're taking in a lot of liquids, your kidneys can reabsorb less water without the body becoming dehydrated, right? The kidneys are trying to balance water intake with water output. So the more water that you take in, the more water can be released as urine, right? So if you're drinking lots of water like I am now, there's very little, if any, antidiuretic hormone that's being produced, and so you will have this. You'll produce lots of urine, and that urine will be relatively clear, dilute, right? All of us have that experience, right? Drink lots of liquids, you urinate a lot, and it's clear. What if, on the contrary, you are not drinking a lot of water, either because you're busy or you forgot your water or because you've been sleeping all night, right? Most of us don't drink while we're sleeping. Well, to prevent dehydration, the posterior pituitary gland produces antidiuretic hormone. What does it do? It causes a portion of the distal convoluted tubule and the collecting ducts to reabsorb water. And so you end up with a smaller volume of urine, and that urine will tend to be more concentrated. Because it's more concentrated, it will tend to be darker in color. That's why if you slept all night long, you get up and you go to urinate, the urine will tend to be darker in color. Also, if you're not drinking sufficient amounts of water during the day, you don't produce as much urine, and then when you do go and produce urine, you can tell it tends to be darker color. Right? So this is just a chart that I pulled off of a place called nursebuff.com. I do not know, to tell the truth, how accurate these descriptions are until when your health is at risk versus when it's not. I use this more just to show you, to illustrate how different in color your urine can be. The lighter it is, the more hydrated you are. The darker it is, the less hydrated you are. That's all the impact that antidiuretic hormone is having on you. If you ever drank alcoholic beverages, you will tend to have a lighter colored urine because alcohol suppresses the production of antidiuretic hormone, and that is also why, for the most part, if you do a lot of drinking before you go to sleep, the next morning you have that the day after effect, also called the hangover. It's mainly dehydration, because when you should have had ADH causing reabsorption of water by the kidneys at night, you woke up in the morning, went to urinate, and instead of your urine being dark, it was clear, and so basically you slowly dehydrated during the night. Okay, any questions about that? So the last topic I want to cover, and I do realize I'm over my time. If you have to go, I completely understand it. This is being taped, so you can always go back and look at this. But so many of you had questions about pH that I do want to talk about, and it is important. So this will be the last topic that I talk about, and then I'll open up the floor for questions if you have any. Okay, so pH of blood. Normal range is 7.35 to 7.45. If you go out of that range, if you're above 7.45, it's considered an alkalosis because it's more alkaline or basic than it should be. If you're below 7.35, then it is considered an acidosis, even though technically this whole range is above 7. So it's all alkaline, but because it's relative to the normal range, it's an alkalosis if it's above 7.45 and acidosis if it's below 7.35. Okay. Now, your body naturally produces hydrogen ions. It's just a regular, normal byproduct of cellular metabolism. So your body has to have a mechanism for regulating, or mechanisms for regulating pH, because it normally produces substances that's going to move the pH of the body out of that normal range. Just a reminder, pH is simply a measure of the concentration of hydrogen ions. In fact, pH literally means potential of hydrogen. Here's the actual equation right here. What's important to understand is that the greater the hydrogen ion concentration, the lower the pH. And the pH scale is a logarithmic scale, means every unit is a 10-fold change. So something with a pH of 6 has 10 times the hydrogen ion concentration as something with a pH of 7. 7 is neutral. Above 7 below 7 is acidic. Above 7 is alkaline. And the farther you get away from 7, the more acidic or alkaline the substance becomes. Okay? Why do we care about pH? We care about pH because if pH moves too far out of the normal range, it disrupts hydrogen bonds and it changes the shape of proteins. And proteins are what do work inside of the body. Proteins act as enzymes. Proteins can transport substances across the plasma membrane, right? Things that are doing things in our body, substances that are doing things in our body in order for it to function are the proteins. And their function is based on their shape. Change the shape of a protein is like changing the shape of a key. Will a key work if you change its shape? No, right? So in this example, here's an enzyme. That enzyme shape is required for its function. These little arrows are showing you where there are hydrogen bonds between different amino acids. If we have a pH change that breaks those hydrogen bonds, it completely unravels that protein. Now that protein doesn't work anymore. So that's why pH is important. Okay? We do have what's called absolute low and high for pH, where if you get outside that range, death will occur. And it's mainly because of the effect on the nervous system. But a pH below 7 or pH above 7.8 will lead to death. But even before you get to those absolute numbers, there can be significant effects on normal functioning of the body and set the body up for disease. So pH is very important. Okay. So we have 3 basic mechanisms to help maintain normal blood pH. We have what are called chemical buffers, we have the respiratory system, and we have the kidneys. Okay. Just so you, as a review, acids are proton donors. You put them in water, they release hydrogen ions. Bases are proton acceptors. You put them in water, and they release substances that will then pluck hydrogen ions out of solution. Let's look at a strong acid as an example. So a strong acid would be HCl. You put HCl in water, it dissociates into hydrogen ions and chloride ions, and it does it pretty much completely. Like you won't find any HCl as a molecule in the solution. It'll be all hydrogen ions and chloride ions, right? And these hydrogen ions are what do damage to clothing or tissues or anything else, because they'll break bonds trying to become stable, right? That's why you wouldn't want to consume hydrochloric acid. Weak acids don't dissociate very much. They mainly, you put them in water and they remain intact. They don't dissociate much into hydrogen ions and bicarbonate ions. Okay, in this case, right? They don't release very many hydrogen ions. Most of it stays as an intact molecule. Carbonic acid, by the way, is soda pop, right? You bubble carbon dioxide through water and you get carbonic acid. You can drink carbonic acid. Why? There are very few hydrogen ions, free hydrogen ions moving around. Okay, so that's the difference between a strong acid and a weak acid. Same thing with a strong base versus a weak base. You put a strong base in water and most of it's going to dissociate or ionize, right? Sodium hydroxide is a strong base. It's found in oven cleaner, right? These hydroxide ions do a very good job of breaking down other molecules, right? That's why oven cleaner is a powerful cleaner, but it can also break down your own tissues, which is why you never spray and you never want to have direct contact with oven cleaner, right? You always wear gloves or something. Strong acid. Weak, that was a strong base. A weak base, an example would be sodium bicarbonate, also known as baking soda, right? You can eat baking soda. It's in toothpaste or some toothpastes anyway, right? Why is it a weak base? Well, you put it in water and most of it stays the intact molecule. A little bit of it will dissociate into sodium and bicarbonate, but not much. All right. Now, why am I talking about weak acids and weak bases? Well, first of all, so you understand what makes a weak acid, a strong acid or base, strong acid or base, but also because weak acids and bases make great what are called buffers. Buffers resist changes in pH, and your body has several what are called buffer systems. Now, to sort of illustrate how buffers resist changes in pH, let's look at the following sort of, at least I think I have a, oh, cool, yes. All right, little illustration here. So buffers resist pH changes by taking up excess hydrogen ions or releasing hydrogen ions when there's a deficiency. So pH is concentration of hydrogen ions. Let's say these two hydrogen ions here represent the normal range, right? So this is the normal range. This is a buffer here on the left. Let's say there is this production of excess hydrogen ions. How will that affect pH? Well, if we go from like 2.5 times the normal concentration of hydrogen ions, that's going to really drop blood pH. How does a buffer help resist that? Well, the buffer takes up excess hydrogen ions, right? And therefore helps restore the pH to its normal range. Now, it's not quite where it was, but it's much closer to where it was than it would be if the buffer is not there. So that buffer has taken up excess hydrogen ions. Now, let's say a base is introduced into the body, right, which plucks those hydrogen ions out of solution. Well, now the buffer releases hydrogen ions to try to, again, resist that change in pH. So a buffer takes up hydrogen ions when there are excess numbers of them and releases hydrogen ions when there's a deficiency of them. And that way, buffers help resist changes in pH. The human body has 3 main buffer systems to do this. It has a bicarbonate buffer system, a phosphate buffer system, and a protein buffer system. And that's all I want you to know. I want you to know them by name. In terms of strength, know them in order. Like the bicarbonate buffer system is the strongest, is the weakest. The protein buffer system is the strongest. Now, now these are slides that sort of illustrate them, but you don't really have to know this in detail. What I basically try to show here is that when you have a strong acid interacting with a weak base, it converts that strong acid to a weak base, converts that strong acid to a weak base, and that helps minimize changes in pH. If you have a strong base interacting with a weak acid of the bicarbonate buffer system, it converts that strong base to a weak base. So the bicarbonate buffer system, again, is minimizing changes in pH by, in this case, converting strong acids and base into weak acids and bases, right? Same thing with the phosphate buffer system. I won't go through the details because, again, you don't have to know them. I'm just trying to illustrate how these buffer systems work. They convert strong acids and bases into weak acids and bases, okay? And your proteins do something similar, right? They take up hydrogen ions when the pH is, they release hydrogen ions when the pH is high. They take up hydrogen ions when the pH is low, all right? So that's how buffer systems work. But in general, they're not very strong, right? And they're limited. Your respiratory system also acts as a buffer because it can remove more or less carbon dioxide. Your respiratory system is more powerful in terms of controlling pH than all the buffer systems put together. And the way it does it is it controls the amount of carbon dioxide removed from the blood. So this is a very important equation. Okay, you will want to commit this equation to memory. Carbon dioxide plus water produces or yields carbonic acid, which then dissociates into hydrogen ions and bicarbonate. Where does the carbon dioxide in your blood come from? Well, it comes from the tissues that release it, right? The tissues produce carbon dioxide all the time. That carbon dioxide then combines with water in your plasma, forms carbonic acid, and voila, it dissociates into hydrogen ions and bicarbonate ions. Okay? In the respiratory system, when we were covering, we said that the higher the carbon dioxide levels in the blood, the more acidic the blood, right? And you understand why. The more carbon dioxide you have, the more hydrogen ions you have, the lower the pH, the more acidic it is. So your respiratory system can regulate blood pH by removing more or less carbon dioxide. If blood pH falls, it can remove more. If blood pH increases, it can remove less. And that's how it works. Okay? So how do the lungs respond if there's an increase in blood pH? Let's say you all of a sudden you had a really bad stomach ache and you ate like a whole huge amount of Tums, right? Because you were in such pain. That's going to flood your system with, Tums are an antacid, right? They're a base. So they're going to tend to increase your blood pH, and it's going to tend to cause a drop in the hydrogen ion concentration in your blood. How's the respiratory system going to respond, right? Fewer breaths, and those breaths will be shallow. The lungs will remove less carbon dioxide. That will tend to increase carbon dioxide in the blood, carbon dioxide, and then hydrogen ions in the blood, bringing blood pH back to the normal range, or at least it will try. What about the opposite? What if there's a decrease in blood pH, right? Let's say you drank there was a sale on Coke and you just drank a whole bunch of Coca-Cola, right? Maybe you took out two liters, and now your blood pH is decreasing, meaning the hydrogen ion concentration is increasing. Your lungs will respond by attempting to remove more carbon dioxide. So your respiration rate is going to be higher and deeper. Okay? Now your lungs do this all the time without you having conscious knowledge of it, right? As pH increases and decreases on the daily basis, on an hourly basis, your lungs respond by increasing and decreasing how much carbon dioxide it removes, right? Now, obviously, there's a limit to how much the respiratory system can modify your pH, right? Okay, lungs can vary carbon dioxide levels, but they can't replenish bicarbonate, and they can't remove all the carbon dioxide, right? There's a limit, okay? The kidneys, however, they're more powerful than the lungs because they can actually remove the actual acids, or they can actually make more base. They're slowest to react, but they are the most powerful because they can literally remove the acid from the body or remove the base. So let's bring the kidneys into this equation. So how do the kidneys come into this? Well, they can literally remove more hydrogen ions or less and make more bicarbonate ions or less, right? So if your blood decreases, I'm going to go back one. Let's say the pH of your blood decreases. Let's say, for example, you have emphysema, or you have some kind of respiratory disease that doesn't allow your lungs to remove the normal amounts of carbon dioxide. Well, the kidneys that will cause an increase in hydrogen ion concentration. Well, your kidneys can literally remove more hydrogen ions from your blood and excrete it as urine, right? The kidneys can also generate more bicarbonate. So that's how you can have someone who has very bad emphysema or very bad bronchitis, elevated levels of carbon dioxide, but their blood pH is normal. Why? Well, because the kidneys are compensating by removing more hydrogen ions and producing more bicarbonate. Consequently, that's going to significantly change the pH of the blood, right? I'd expect someone who has some kind of significant respiratory disease, but yet has a normal blood pH, so good functioning kidneys, to have a really low blood pH because the kidneys are going to be really pulling out a lot of hydrogen ions and putting it into the blood, into the urine, excuse me. And on the flip side, what if the pH of the blood increases. Again, let's say maybe, I don't know, the person was consuming something that was very alkaline. Well, the kidneys could remove less hydrogen ions and remove more bicarbonate to maintain the normal range of blood pH. All right? So the kidneys are very powerful in terms of regulating blood pH. And it's also why you can have very relatively wide ranges in blood pH depending on the individual's diet and depending on other circumstances. Okay? Urine pH, for the most part, can vary 4.5 to 8. This is not to say it can't vary from this range, but this is the normal range as provided by your book. So I would say probably 95, under normal circumstances, 95% of individuals would fall in this range. Okay? All right? So those are the 3 mechanisms that the body has to regulate blood pH. Those chemical buffers, the respiratory system, and the kidneys. Okay? Key to understanding that is this equation right here. Carbon dioxide plus water yields carbonic acid, which yields hydrogen ions and bicarbonates. Commit this equation to memory. Understand how, as carbon dioxide levels rise and fall, hydrogen ions rise and fall, therefore causing the blood to become more or less acidic, and then where the respiratory system and the urinary system impact this equation. Okay, so with that, I'd like to open up the floor for any questions you may have.